Thank you everyone for attending this webinar. In this talk, we will discuss the concept and the application of in vivo delivery for CAR-T cell therapy. Let's first start with what is in vivo CAR-T. Cell therapy has become an effective treatment for oncology in recent years. However, the extremely high cost of a manufacture is a long-lasting bottleneck for the majority of a patient in need. There are people working on off-the-shelf universal CAR-T, but it is still not as potent as the autologous CAR-T. Nowadays, the in vivo gene delivery approaches provide a much more cost-effective way to treat the patient. Instead of preparing the CAR T cell in ex vivo, we simply inject the gene encoding the drug into patients and let the patient's T cell to engulf the gene fragment and express the CAR domain in vivo. By doing this, we can skip the whole complicated process like T cell extraction, transfection, cell expansion, storage, and transportations. The in vivo gene delivery is achieved by two different systems, the viral and the non-viral approach. Lentivirus is the most mainstream platform for viral-based deliveries. It is incorporated into patient's genome for long-term stable expression. The T-cell targeting can be achieved by pseudotyping. However, a virus will trigger significant amounts of immunogenicities and it is subject to rapid clearance. Alternatively, the TLNP delivers mRNA, which is a transient approach for gene expression. The T-cell targeting efficiency is relatively low, but given the lower immunogenicity risk, people can be repeatedly administered for long-term efficacies. For both cases, how to precisely and effectively deliver the CAR gene into T cell become a crucial factor. Under such circumstance, we can add an antibody specific to T cell biomarkers, such as a CD3, to greatly improve the T cell targeting efficiencies. This process is called T cell retargeting. In recent years, both viral and non-viral in vivo CAR-T demonstrate an explosive growth. Although virus-based approach takes roughly 2/3 of the clinical pipelines, the non-viral approach is dominant in preclinical and discovery stage. This is the overview of a workflow from discovery to CMC for these two approaches. Either one requires the discovery of two antibodies. One is the CAR lead antibody, which targets the CAR T cell to cancer cell, while the other one is the T cell targeting antibody, which targets the gene fragments into T cell, so that the T cell can express the CAR on the cell surface. Lentivirus approach is relatively more straightforward. Everything is incorporated into one module with the gene sequence encoding different parts. For the LNP approach, the discovery of LNP and mRNA are independent from those two antibodies. After pharmacology profiling, the final candidates will go through the CMC process. Again, the LVV approach is relatively simple, while the LNP approach consists of three CMCs, one for antibody, one for mRNA LNP, and one for the conjugation process. Now let's dig into the detail of the process, starting with the CAR lead antibody discoveries. ProBio offers a one-stop solution including antibody discoveries by a variety of platforms like hybridoma, phage displays, single B cells, engineering by humanization and affinity maturations, in vitro assays for CAR-T cell function characterizations, and finally the in vivo disease model, including efficacies, PK, and toxicology studies. For the CAR lead antibody generation with the single chain SV format, there are three options. Hybridoma is the most traditional approach, although it takes about 4 months from immunization to lead antibodies. The hybridoma cell lines provides the most flexibility in terms of screening strategies. You can include multiple antigens for multi-dimensional profilings. Single B cell offers a balance between timeline diversity and antibody affinities. It can complete screening against the multiple antigens within 1 day, followed by B cell extraction and further characterizations. Human naive FAB libraries is the most expedited approach. It takes about 2 months, but with the sacrifice of antibody affinities. Alternatively, if you are looking for single domain, the VHH as the CAR module, immunized phage display libraries is the most well-established platforms. In addition to standard screening criteria, the phage panning process can provide feasibilities to enrich for antibodies with unique features like pH dependent bindings. Alpaca naive library is also a viable option with much shorter timeline, and of course it also comes with the sacrifice of antibody affinities. After recovering the candidate sequences, humanization is needed to reduce the overall immunogenicity risk. Typically, it includes sequence design, screenings, and affinity validation by SPR. Additionally, you can also choose to conduct in silico developability assessments, including post-translational modifications, aggregations, and immunogenicities. Humanization is a pretty standard service at ProBio. It usually takes about 3 weeks with a guarantee of affinity retaining as the parental antibody. As I mentioned previously, antibody derived from naive library usually comes with worse affinities. It may need in vitro affinity maturations. Instead of using phage display and combinatorial mutagenesis libraries, ProBio adopts a unique strategies. After the AI-aided modeling and mutation design, we will select a few hotspots within the CDR. A precise site saturation mutagenesis library will be created for each of those hotspots in the CDRs. Then a E. coli-based expression system called FASIVA will be used to express this mutation library, and the supernatant from E. coli monoclones will be collected for the SPR affinity rankings. Based on the off-rate data from these PML saturation mutation libraries, the single residue beneficial mutation will be isolated and built into a second combinatorial mutation library for further affinity assessments. As you can see from the data of these case studies at the bottom, with this strategy, we're able to improve the affinities by hundreds, even thousands of fold to achieve the picomolar affinity range. After finalizing the CAR antibody sequence, the CAR T cell will be generated, followed by functional screenings with a panel of indicators, including the T cell surface expression levels, the cytotoxicities, the cytokine release levels, and the T cell proliferations. Cytotoxicity can be evaluated by two different approaches. Reporter assays is widely used for early stage screenings, while the primary cell killing assays is preferred for confirmation and profiling. Comparing to IO therapy, the balance between efficacy and safety is the top priority for cell therapies, especially CAR-T therapy. A number of factors can be considered during the CAR antibody discoveries. The epitope and the affinity of CAR antibodies is the first driving force of toxicities. The design of the CAR structure can further modulate the safety profile. The on-target off-tumor effect can be alleviated by leveraging the avidity versus affinities. On the other hand, bispecific CAR and combination therapy with IO are also popular solutions to improve the efficacies without jeopardizing the toxicities. Now, let's move on to the T cell targeting antibody discoveries. As I mentioned previously, the T cell targeting antibody is another crucial module for the in vivo CAR-T delivery systems. So here is a summary table of some clinical and preclinical pipelines. The typical T cell targeting antigens including CD3, CD7, CD8, etc. For the generation of these targeting antibodies, there are two different options. Ready-to-use antibody from published literatures or de novo discovery of a novel sequence. The typical timeline from material generation to pharmacology validation is 3 and 8 months, respectively. The general process of de novo discovery for the T cell targeting antibody is pretty much the same as the CAR lead antibodies as we previously discussed. So now let's focus on the ready-to-use approach. SP34 is a well-characterized CD3 antibodies widely used in a variety of clinical therapeutic antibodies, such as a T-cell engager. It is originally published in a paper which makes it available for everyone. You can use it without paying for the IP. Published data indicated that SP34 can effectively target the in vivo CAR-T deliveries. So on the right panel, the peak of the CAR-T cells after the infusion demonstrates successful incorporation of a CAR gene into the T cell. While on the left panel, the depletion of a B cell after infusion suggests that the B cells are killed by the anti-CD20 CAR-T cell, which also implies the successful incorporation of a CAR gene into the T cell. SB34 is a mouse-derived antibody, and it cannot be directly used as therapeutics. Our team did humanization and generated a panel of 18 humanized SB34 variants with diversified germline and roughly 5-fold affinity range. You can choose from this toolbox for in vivo CAR-T validation, skipping the timeline and the risk of the humanization process. All right. After the identification of CAR and the T-cell targeting antibodies, we now move forward to the pharmacology validation of the in vivo deliveries. First, for the in vitro part, we have talked about the CAR lead functional validation in previous sections. For the evaluation of the in vivo deliveries, the bioassay is a little bit different. We use either Jacka cell or PBMC to measure the infectious titer to determine the T cell targeting efficiencies. In this case study, we're using the LVV lentivirus system to deliver two genes, the GFP and the CD19 CARs genes on the left and the right panels respectively, through antibodies against two different T-cell biomarker A and B. On both cases, we observed significant expression of GFP and CD19 CAR on the wild-type Jacka cell in a dose-dependent manner, while there is much lower level of expression when the T cell biomarker A or B is knocked out. So this data demonstrated that the targeting antibody against the T cell biomarker A or B will effectively improve the efficiency of in vivo gene delivery. In another case study, we are utilizing the PBMC instead of a Jacka cell to evaluate the delivery efficiencies. The GFP gene was delivered in vivo by LVV, the lentivirus system, with targeting antibodies against T-cell biomarker A or B on the left and the right panels respectively. So on the top left panels, the x-axis is the expression of GFP, the gene to be delivered, while the y-axis is the expression level of the T-cell biomarker A. After transfection of the LVV with escalated dose from 0 to 80 MOI, we observe enrichment of cells in the Q2 quadrant, which is double positive for both GFP and the biomarker A. In the meantime, there is not much change in the Q3 quadrant, which is GFP positive but negative for the biomarker A. So this data showed that for those cells with high expression of a biomarker A, the GFP gene is successfully delivered and expressed, while for those cells without the biomarker A, there is not much GFP delivered. It demonstrated that the delivery of a GFP gene highly depends on the presence of a biomarker A through the targeting and through the T-cell targeting antibodies. In addition, There is one more parameters in these case studies. The pre-treatment of a PBMC with anti-CD3, CD28 beads. Unlike the top panel, untreated PBMC showed minimal amount of GFP positive cells, suggesting lower gene delivery efficiencies. The CD3, CD28 beads can activate the T cell, and the difference between top and the bottom panel suggests that activated T cell comes with much higher efficiency for in vivo gene deliveries. And the right panel with the biomarker B basically tells the same stories. Following the in vitro cell-based validation of in vivo deliveries, animal disease models is the next step. Similar to the antibody-based therapy, it also includes three panels: the efficacies, the PK, and the toxicities. For efficacies, we have established multiple models for both oncologies and autoimmune diseases. You can use these models to screen for candidates or to study the dose response kinetics. For PK studies, it includes the evaluation of on and off-target deliveries, the transfection efficiencies, the CAR-T cell in vivo persistency, and the infiltration. The toxicity studies covers the TCR profilings, the cytokine release, the general toxicity, and the pathologies. For oncology, we have established disease models for 24 tumor indications with more than 80 tumor cell lines. In addition, there is a cell bank with more than 200 tumor cell lines for customized assay development. Similar to in vitro assay, the evaluation of in vivo delivery by animal disease models is different from the simple tumor growth inhibitions. Here, I'm sharing a case study of in vivo gene deliveries by antibody retargeted lentivirus using a human PBMC engrafted mouse models. The LVV encoding CD19 CAR gene is coupled with antibody against a T cell biomarker A or B on the left and the right panel, respectively. In both cases, we observe a time-dependent expression of CD19 CAR in the mouse PBMC after LVV administrations, but no visible change with the vehicle control. This data demonstrate that both A and B retargeting antibodies can effectively deliver the CD19 CAR in vivo. In a separate case study, we did a similar experiment using a different mouse models. Instead of transplanting human PBMC, the endogenous gene of CD19 and the T cell biomarker B were replaced with the human version. Similarly, we observed a time-dependent expression of a CD19 CAR after the lentivirus administration, but no visible change with the vehicle control. This data further demonstrates the specificities and the efficiency of antibody retargeted lentivirus expression systems. All right, at the end, I would like to say a few words about ourselves. First of all, we have extensive experience and the expertise in antibody discovery, facilitating the generation of both the CAR antibody and the gene delivery retargeting antibody. Second, we are a one-stop partner with capabilities from discovery, pharmacology to CMC and the commercial manufacture, and it covers both viral and the non-viral options. Last but not the least, we provide comprehensive disease models for both oncology and autoimmune diseases, and we can support the non-human primate study with our external collaborators. We're truly hoping that a reliable partner like us can help you to accelerate your preclinical pipelines. All right, that's all. Thank you, everyone.